I'm happy to talk today a little bit about something near and dear to my heart, the Bioburden standard. So I'm Martel Winters, work for Nelson Laboratories, and I'm also a U.S. representative to the ISO working groups for the Bioburden document. So I'm excited to be able to talk a little bit about this today. Okay, so just a little bit of background. A couple of years ago, ISO Working Group 2, that's the radiation working group, they came up with a list of items that they wanted Working Group 8, that's the microbiological methods working group, that they wanted us to add to the Bioburden standard. Things that they felt, guidance that they felt was not currently present in the radiation document, the radiation standard. So primarily, the information they were requesting was more guidance, not more. The requirements they felt were sufficient the way they were, but they felt that it would be beneficial to have more guidance in the document, specifically for those involved with sterilization. So I'm going to include some of the main concepts they're going to have us add. Now I don't know, of course, exactly how the text will read when it's done, but I do know some of the main concepts that we'll be adding. Now, you saw I had this PS, what happened to the microbiologists, and my main reason for having that in here is a lot of the guidance they're asking us to add into the Bioburden standard is a result of the fact that we're seeing a reduced quantity of knowledgeable microbiologists that work on site at manufacturers. And it's an obvious trend in the industry that there are not sterilization or manufacturing microbiologists that work on site at the facilities. Now, oftentimes, microbiology and sterilization is given away to someone else. They say, well, while you're at it, you're in QA, so why don't you do microbiology and sterilization? Or you're in RA, or you're in manufacturing, why don't you also handle microbiology and sterilization? And the result is that many medical device companies are then relying heavily on contract laboratories and contract sterilization facilities to provide guidance on how they should do microbiology and sterilization. Now, that's not bad, because those of us that work at contract labs or sterilization facilities, we feel that we know what we're doing, but we're not working at your facility, and so we don't know exactly what is best for you in your situation. So the information you're getting might not always be the right information for your specific situation. And one of the biggest things that we see is that after we have discussions on the phone with the facility, the result of the discussion, the rationales that are used to decide on a certain microbiology test or to decide on a certain validation approach for sterilization, those are not properly documented. And so when later on you're going back, well, why did we choose this? And you're going back trying to find a written rationale, and there's nothing there. So how often have you heard, or how often have you said this, maybe I should say, so someone asks you, okay, why did we choose this? And you go back, well, there's nothing in the files. We don't know why, that it's, we can't find it. So if we think of an analogous situation, how often do we ask someone in RA to also be a project engineer? And say, well, you're a smart person, let's have you engineer the product as well. Or a finance person to also be a QA manager. Now, in startup companies, I know you have 5 people. A lot of people wear a lot of hats. But once you're out of the startup situation, these are not things that you do. So then, why do we ask something critical like sterilization to be one of 20 items on someone's list of job descriptions? And especially someone who has no background in microbiology or sterilization. If you can't tell, this is my soapbox part of my presentation today. So really, is sterilization any less important than engineering or regulatory affairs? I mean, microbiology and sterilization are critical to what we do in the medical device industry. What we're seeing, so as we sit at these, like the AMI sterilization meetings or the ISO sterilization meetings, what we are seeing as people that are attending are sitting around the table or sitting around in the room, is we're having discussions about what the standards should say and how they should say it. We're seeing there is a decrease in expertise in manufacturing microbiology and sterilization microbiology. And it's a direct result of the fact that most companies don't have in-house people they're developing to be very knowledgeable in those topics. Now, my purpose here is not to guilt trip everyone into hiring a microbiologist or a sterilization person, so that's not where I'm going with this. So as a result of having this lack of expertise in the industry, from standards writings bodies, from AMI and ISO, we are trying to add more guidance into the documents to help people understand some of that information that we're lacking. Now, one of our concerns is as we put more guidance in the documents, there is also more information that can either be misunderstood or that can be taken as a requirement rather than guidance. You guys know as well as anyone else, when something's in a document, FDA or other regulatory bodies will often say, well, it's in the document, it's there. I know it says guidance, but we're going to require it anyway. So we're trying to walk this fine balance. So what I'm really saying here is when you are making decisions regarding microbiology and sterilization, which should be occurring in all phases of manufacturing, from product design to the manufacturing process design, sterilization and routine monitoring, those all involve microbiology and sterilization decisions. So as you're going through that, include knowledgeable people in that process. And I understand it's difficult to have a full-time microbiology or sterilization person, but there are knowledgeable people in the industry, so involve them as you're making those decisions. The benefit to involving these people in these processes is that whoever you've assigned microbiology and sterilization to, as they're working with these people, they are beginning to build up that internal expertise. So the long-term benefit is much better. All right, seminars are always good, right? But I say plural there because, we at Nelson Labs, we host seminars around the nation several times a year, and it is extremely common for us to talk to someone, why are you here? And they say, well, I was assigned microbiology and sterilization, so I came to a seminar to learn what I'm supposed to do. And they attend one seminar every 2 or 3 years, and that's enough to become dangerous, but not enough to become really effective. So and also, different seminars will be taught from a different perspective, and so you'll gain much better knowledge. One of the questions that we do frequently ask is, we say, well, here's some guidance that we want that people are asking for, and we as microbiologists, we say, well, this should be obvious. Do we have to add this in there? But so again, back to that balance. How much do we add to be helpful versus let's not add too much to where people think it becomes a requirement and it gets misunderstood. This last one is something we see quite often, where all of a sudden there's a problem, okay, and I get brought in on problems quite often, and the interesting thing is almost every problem I've ever been brought in on, it is not something which just occurred last week. It is a foundational problem that, had it been established properly in the beginning, it would likely not have occurred. It would have been caught earlier on, or it never would have happened at all. All right. So I'll get off my soapbox now and go on to some of these topics that are going to be where we're going to start adding guidance into the bioburden standard. Low bioburden. We're seeing a lot of products with much lower bioburden now. And I think that the reason is twofold. Either one, because there's more automation involved in the process, or there's a higher concern with bioburden. And so people are involving more strict manufacturing processes. Tissue and biologics tends to be fairly clean, usually. And then also the bottom one there, as a lot of newer products are more high-tech. And so you can't just blast it with a normal dose of radiation or a normal cycle of ethylene oxide. Oftentimes we're seeing more products coming in that require more optimized cycles. And as a natural consequence of that, the bioburden must be more strictly controlled. So low bioburden is not a problem necessarily, but what we see is that if your product has low bioburden, or if you need your product to have low bioburden, there are some issues that you have to deal with. Often you're looking at different bioburden test methods. The methods that we have used forever often need to be tweaked a little bit to work better for your situation. A few examples here: either instead of testing one product at a time for bioburden, you might need to pool multiple samples together so you can get a larger sample size, so you can get your limit of detection down lower. Most probable number, or MPN testing, is something that's not done very often, but it's a very good test method for certain types of products. Or maybe you're taking your typical bioburden test method, but you need it to be more sensitive, so that you can resolve down to 1 CFU or 2 CFU. Now, another problem with low bioburden is you often have to evaluate the data differently. And I'll go more into that in just a little bit. But the way that you look at the bioburden data you obtain is often changes when you have low bioburden. And the last thing, also, you can see larger swings. When you have a bioburden that tends to be 0 CFU or 1 CFU, you can see larger swings in bioburden count. So the occasional bioburden spike, or maybe not occasional, but the frequent bioburden spike. And these larger swings in bioburden, it can have a significant implication for some types of sterilized products, because it demonstrates a lack of control in the bioburden in the manufacturing process. Okay, so I'm going to go a little more in depth in a few of these. Sample pooling, this is a great option for a manufacturer, because instead of testing 10 samples, I need to stop spitting into the microphone here. Instead of testing 10 samples in individual tests, you're putting them all into one test. So financially, that sounds great. You're only paying for one bioburden test rather than 10. Now, the advantage, another advantage, it does give you a lower limit of detection. So instead, those of you that are familiar with bioburden data, if you're getting lots of zeros, you're used to seeing a less than number probably. Less than 2, less than 3, less than 10, depending on what portion is being tested. But pooling samples together, you can resolve down to less than 1, like 0.1 CFU is your limit of detection when you pool samples together. The big problem with pooling is that it will mask individual bioburden spikes. So an example here, let's say you pool 5 samples together, and the result of your 5 samples pooled is 4 CFU. So mathematically, you just divide 4 CFU by 5 samples, so your average is 0.8 CFU. Now, if all 4 CFU came from one sample, rather than them being split up between the 5 samples, it's less of an issue with 4. But if you see 100 CFU, then it can be a big deal for you whether each sample had 20 CFU or whether one sample had 100 and the rest had zeros. So that's one of the biggest issues of sample pooling, is you lose the ability to understand your bioburden variation in the sample set. So here's some test data. Prior to going to a pooling scenario in bioburden testing, we always recommend that you do some testing separately first. So if you're doing testing separately, you got your aerobic bacteria column, you see mostly you're getting zeros, occasionally a few CFU. The fungi, mostly zeros again. So you see that the bioburden range is very low. You're not seeing a large range in bioburden. So this would be a situation where the bioburden testing, pooling these samples together would be a viable option. Now, if you're seeing data more like this, then it really is something you should consider keeping testing separately. You gain no advantage to pooling. You gain a financial advantage, but your quality advantage, you actually lose data here. Where you're seeing the occasional bioburden spike, you got a 56, 120, and if you were to pool these together, you would have no idea that you're getting that level of bioburden variation. All right, so increasing sensitivity in the bioburden count. Mainly what we're talking about here are less than values, and this will be something we talk about quite a bit as well in the new version of the bioburden standard. Is those of you that have very low bioburden, I'm sure you're used to seeing less. Hopefully you don't see 0. You should never see 0 in a bioburden report. Hopefully what you're seeing is less than something, less than 2, less than 4, less than 10. And when your bioburden tends to be lower, then it will be better if you have a more sensitive bioburden test. A few ways that we do that, because we don't want to see less than 4s. If you're getting a lot of less than 4s, then you really should have a more sensitive bioburden test. Less than 1, less than 2, that makes more sense. So a few ways that we do that. One is what we call a volume factor. In most cases, in a bioburden test, you're doing an extraction followed by filtration. And usually you might be putting in, for example, 400 milliliters. You might only be filtering 100, and that's why you have that volume factor of 4. And that's why you're getting a less than 4 on your report. So the simple thing is just filter more of the extraction solution if you can. If you want to get less than 1, get down to less than 1, you can filter all the solution, and once you put that membrane filter on a plate, you incubate it at one temperature, do a count, incubate it at another temperature, and then get your fungal count. So that's a common way to do that to keep your factor down. If you want less than 2, you just filter half the solution through one filter for your bacteria, the other half the solution for a separate filter for your fungi, and then incubate those at 2 different temperatures. An SIP is not something that's done very often, but sometimes that can bump up, that can really decrease the sensitivity of a test. If you're only testing 10% of your product, then no matter how much of the solution you're filtering, you can't resolve any better than 10. So if you're getting 0s, you're going to get less than 10 CFU. So if you're testing an SIP, it's easy to test a larger portion of your device. And then pooling samples can also give you better sensitivity, and I've gone through that already. There are actually many other options as well. These are the ones that kind of float to the top as being most common, but depending on the situation, there are other options to consider. So how low is low? This is something else we'll be talking about in the standard. As you know, there is no bioburden value at which you pass. There's no pass-fail for bioburden testing in most situations. The sterilization method does help to dictate what you would consider low. In a bioburden-based method like radiation, low might be one thing. In an overkill type method like EO or steam, then low will mean something very different. Now radiation, this is possibly something, we talked about this at the ISO meetings a couple of weeks ago. We'll likely be putting in some guidance there that low might be less than 10 CFU, and a new term of ultra low would be less than 1 CFU. There are some rationales behind that based on the radiation sterilization science that I won't get into, but that might come out. We talked about that as an option. Now, low bioburden, what repercussions does that have speaking for radiation? Radiation is a bioburden-based method, so your radiation dose is tied quite closely to your bioburden count on your product. So that means for radiation, you might have to test more samples initially than the normal data sample set, and you might have to test more often if you have low bioburden, so that you can prove maintenance of that low bioburden until you gather a really good history. All right, so bioburden test methods. This is probably one of the most common test methods that we do. Well, it's definitely the most common test method we do at the lab. You're cutting up the product, disassembling, immersing it in usually a peptone tween solution. You're extracting using some kind of mechanical shaking. Then you're doing a membrane filtration through a 0.45 micron cellulose filter. And then this filter is being placed on TSA and or SDA for fungi. And then you've got those two different incubation conditions. Now, the question that we're asking, and this goes back to having fewer microbiologists involved in these processes. If you call a lab and say, what should I do for my bioburden test? What I just explained, that's the answer you're probably going to get. This is what everyone else does, this is what you should do too. That is not always the right decision for your product. More often we find when we dig into a manufacturer's process or a manufacturer's raw materials or components, we often find that there are different things that we should be doing which would be better suited for your situation. And that's related to those bottom two bullet points there where how often does bioburden testing have to be repeated? So we get samples all the time. We say, okay, well, this makes sense. And the manufacturer says, well, yeah, it makes sense to us. Just do what you normally do. And then we do it once only to find out that something doesn't come out quite right with the test. So we have to go back and repeat testing. So when we are setting up a bioburden test method, and this is something that at Nelson we're trying to do more often. We're trying to make sure that we dig into some specifics regarding your situation to help you better establish what the right test method is for bioburden. So things to keep in mind. What types of microorganisms might be on your materials, your raw materials, or your components? And in order to do that, we have to understand your manufacturing process. We need to understand the origin of all of your materials. And also, where are your microorganisms? Based on the origin, where might the microorganisms be hiding on your product? What potential microorganisms might we find in your assembly process? And so we look at things like personnel and process. We're looking at, is there water involved in the process? Either at your location or at one of your suppliers, is there water there? How are your materials and components stored? And what do your cleaning processes look like? I've seen most of the cleaning processes I've seen actually add bioburden rather than take it away. People have this idea, well, I get these stuff from my supplier, I need to wash them off in some water and do some cleaning there, and in most cases they're actually counterproductive. And another question to ask is which microorganisms are of greatest concern for your sterilization process? And those are things to keep in mind as you are establishing your bioburden test method. Evaluating bioburden data. So we're going to be talking about giving more details on like TNTC, too numerous to count. How do you deal with those? Talk about bioburden spikes, providing maybe some options on how to count bioburden plates that have really high counts on them. Talk about what can cause them. We've been trying for years to have an industry accepted definition for a bioburden spike, and we're gonna attempt to do that again. And then plates that have spreading microorganisms on them, how do you deal with those? And also provide more guidance on zero CFU results, less than numbers, which we've already talked about. So here's an example of a bioburden spike, and hopefully we'll be able to put together some ideas for the standard. How do you deal with this? Do you throw it out as an outlier? Do you include it? And actually, either situation is appropriate depending on what you're doing with the data. So a few ideas on definitions here. The top one there is traditionally been the unofficial definition based on how we select bioburden data in a radiation validation. At Nelson Labs, we published a paper recently with a statistician from FDA where we found that in looking at a bioburden data set, the bioburden data that had one value, which was over the mean plus 2 standard deviations, that one value is what made the data not normal, as opposed to being a normal distribution. So you might kick that around as an option, or based on your internal alert and action levels. And that might be the best situation, because that's a very individualized situation for your product, where you decide for yourselves what a bioburden spike is. Okay, so here's a bioburden spreader. So again, these are the questions we ask. Do we throw it out? Do we assign a value? Do we do additional testing? Identify the bug, investigate. This is the type of guidance that we'll be adding into the standard. And the same questions for a TNTC result. So these are the plates that we don't like to see in a lab. These and the plate I showed you previously, because these are hard to deal with. Okay, so that's the bulk of what we'll be adding into the bioburden standard, at least some of the key points. I am available for questions afterwards. I'll be at the booth as well today and tomorrow. And that's it.